Okay, welcome back my dear students. In this video, I explain about the functions of the ribosomes. So here the ribosomes are responsible in the synthesis of proteins. Okay, I explain about the mechanism of the protein synthesis by ribosomes. Okay, right. First we know the transcription. So what is transcription? So generally, if you take the DNA, if you take the cell, so this is one of the cell, and this is the nucleus, and this one is the nucleus. So nucleus generally contains DNA. Okay, so generally contains DNA. Okay, so here the DNA transcribes the mRNA. So DNA transcribes the mRNA. The template of the DNA transcribes the mRNA. That is process is called as transcription. Okay, so transcription means DNA transcribes to mRNA. The mRNA comes from the DNA. That is called as transcription. So this is the mRNA chain. Okay, mRNA chain. So generally the mRNA chain contains the nucleotides. So three nucleotides. They are called as triplet codons. Okay, which contains genetic information of synthesis of specific protein or amino acid. Okay, contains the genetic information to produce specific amino acid. Okay, so these nucleotides generally have on the mRNA. So mRNA have the nucleotides of the DNA. Generally the DNA gives rise to triplet codons to mRNA to produce the specific amino acid. Okay, this is the importance of mRNA. So mRNA helps in the protein synthesis. That process is called as translation. Okay, so transcription means DNA transcribes the mRNA, that is transcription, and mRNA produces the specific protein, that is called translation. Okay, so after the transcription, generally mRNA comes from the cytoplasm. mRNA comes from the nucleus to cytoplasm. It comes to the cytoplasm. Okay, right. Here in the cytoplasm, there is ribosomes. So they are found free in the cytoplasm. Okay, they are remain separate. The subunits of the ribosomes remain separate in the cytoplasm. Okay, now we know the mechanism of the protein synthesis. Here this is the cytoplasm. If you take this is the cytoplasm, then in the cytoplasm generally there are two subunits. So this is the smaller subunit and this one is the larger subunit. So both subunits are remain separate in the cytoplasm. So when they want to produce a specific protein, then these two subunits assemble to each other. Okay, based on the concentration of magnesium and 0.001 molar magnesium ions. Okay, so this concentration from this concentration, both subunits are assembles each other. Both subunits are assembles each other. Okay, right. So here this is the smaller unit and this one is the larger unit. Okay, so smaller unit has a specific mRNA binding site. So this is the specific mRNA binding site. So smaller unit contains specific mRNA binding site. Okay, so this is the specific mRNA binding site. Okay, so this one is the mRNA. Okay, right. So here larger unit also contains two main sites. So two main sites. So that is two main sites. So that is A site. So that is A site and P site. A site means receptor site and P site means peptide site. The peptide bonds which are formed. Okay. There is another site is also there. That is empty site. So that one is the empty site. E site. That means empty site. Okay. So there is two main sites, A site and P site. Okay. Right. If in this process, so generally the tRNA comes and joins with these structures. Okay. So here the tRNA generally produced by the DNA. Okay, DNA produces the tRNA. So tRNA also comes into the cytoplasm and binds with the mRNA and ribosomes. Okay, right. Already you know the structure of tRNA. Generally tRNA, if you take the tRNA structure like this, so this is the structure of tRNA. This is the structure of tRNA. So tRNA is a clover leaf-like model. Okay, like it has 4 specific sites. Okay, 4 specific sites. So one is this one is the anticodon site. So anticodon site, and this is the ribosomal recognition site. So ribosomal recognition site, and this one is the amino acid recognition site, and here this one is the amino acid binding site. Amino acid binding site. Okay, so like these 4 sites are present on the tRNA. Now this tRNA will bind in the ribosome and mRNA. So generally here in the P site, generally in the P site, the mRNA contains 3 nucleotides. mRNA contains 3 nucleotides, that is A, U, G. Okay, in P site mRNA contains 3 nucleotides. This is called as triplet codon. Okay, codon means it contains genetic information. That's why we are calling the codon. Okay, here the AUC, and here A site there is 3 nucleotides, that is CCC. Okay, right. If now the tRNA comes to binds the first triplet codon AUC. Here, okay, here I draw the diagram. So this is the tRNA. This is the first tRNA which binds. This is the first tRNA which binds to the first triplet codon of the mRNA AUG. Okay, here this is the site of the anticodon. So there is a complementary codons are formed in this site. So complementary codons against of the AUG. Okay, the complementary codons are formed against AUG. That means here there is A. Here the A is binds with the U. Okay, there is not T because it is a RNA. So if one is the RNA, so there is a T replaced by the U, uracil. Okay, thymine is an oxide. Okay, so here U, that means here is A. So and C binds with G binds with C. Okay, like this complementary codons are bound here. Okay, right. So when the anticodons will be already binds with the AUZ. Okay, so then first amino acid will be attached in this site. So this is the amino acid binding site. Here the first amino acid will be formed. Okay, attached. So that is called as methionine. That is generally called as methionine. So this AUZ, that's why AUZ is known as the start codon. Okay, it gives rise the first amino acid, that is methionine. Okay, right. And the second tRNA comes and binds with the second tRNA comes and binds with the binds on the A side of the mRNA triplet codons. Okay, so this is the second tRNA comes and binds on the mRNA triplet codons. Okay, on the A side. Okay, here also there is anticodons are formed. Okay, through binding the CCC. That means here G, and G. So this is the anticodons. Okay, so they combine, they paired. Okay, so after that, then the second amino acid is formed here, attached here. Okay, right. So second amino acid attached here. Okay, like this. So this happens in this process first. Okay, right, in the B site and A site. There are 2 amino acids, two tRNAs are attached with the mRNA and forms 2 amino acids. Okay, so after that, later the ribosome will moves on the mRNA. Ribosomes moves on the mRNA. Okay, so when the ribosomes are moves on the mRNA, then the ribosomes are moves in this direction. In this direction on the mRNA. mRNA not moves, only ribosomes are moves. So when it moves, then the tRNA moves like this direction. tRNA moves like this direction. mRNA moves that direction and tRNA moves the opposite direction of the ribosome. Okay, so then the new tRNAs are joined with the mRNA. So later I explain another video.